After looking at the different cellular functions of carbohydrates, now we move to the classification of monosaccharides. Monosaccharides are also called as simple sugars. They are aldehyde or ketone derivatives of polyhydroxy alcohols containing at least 3 carbon atoms. That is, they contain an aldehyde or a ketone group in addition to hydroxyl groups on them. This figure shows one of the simplest monosaccharide, which is glyceraldehyde. It is a 3-carbon compound in which carbon 2 is chiral. Let us now briefly talk about the configuration of atoms and to describe the chiral carbon of monosaccharides. Consider the following example. A conical flat is symmetrical. If we have a conical flat, and we look at it by using an imaginary mirror as shown in the figure, the mirror image will be exactly the same as the original flask. Hence the flask and its mirror image both are superimposable. Such substances having symmetry and superimposable mirror image are called as achiral. Let us now see the second example. The figure shows left hand. The mirror image of this hand will be the right hand. Now if we try to superimpose the left hand onto its mirror image, which is the right hand, they will not align completely as we have seen in case of the flask. This is because unlike the flask, the hand is asymmetrical. So it is not superimposable over its mirror image. Such substances are called as chiral. Now let us look at the atoms of a molecule. In this figure, a molecule labeled as C is shown. The molecule has a central carbon atom. To which 2 different groups are attached, represented in blue and green color. However, the other 2 groups attached to this carbon atom are identical, both represented in gray color. Now if we look at this molecule C through a mirror, the image obtained is shown as molecule D. If molecule C is flipped and placed over molecule D, it will be perfectly aligned as shown in the figure. So the central carbon atom in molecule C is achiral. Let us now see another molecule labeled as A. Molecule A also has a central carbon atom, but the 4 groups attached to this carbon are all different, which are represented in red, gray, blue and green color. If we look at the mirror image of molecule A and try to superimpose them, we can see that they will not properly align with each other. As you can see that the green and blue groups are not superimposed. This is because the central carbon atom is asymmetrical. So if a carbon atom has all 4 different groups attached to it, it does not have any symmetry and hence is called as chiral carbon. This figure is showing a molecule of glyceraldehyde with central chiral carbon atom with all 4 different groups attached to it. So monosaccharides contain chiral carbons in them. Based upon the configuration or arrangement of groups around the chiral carbon, the monosaccharides are designated as the D form or the L form. Glyceraldehyde has 3 carbon atoms in it. Carbon 1 is the aldehyde group and is not chiral. Carbon 2 has all different groups attached to it and hence is chiral. Carbon 3 has 2 hydrogens attached to it, same atoms, so it is also achiral. Based upon the configuration of groups around carbon 2, which is the chiral center, there are two possibilities. If we look through the central axis of the molecule and the hydroxyl group on carbon 2 projects to the right side, it is called as D-glyceraldehyde. But if this hydroxyl project to the left side of carbon 2, it is designated as L-glyceraldehyde. The D and L forms are distinct from each other in terms of their structure and properties. Some monosaccharides can have more than chiral carbons in them. Let us consider the example of glucose. Glucose has 6 carbon atoms in it. Carbon 1 is aldehyde and is not chiral. Carbon atoms 2, 3, 4 and 5 are all chiral. Carbon 6 is also not chiral. In those monosaccharides which have more than 1 chiral carbons in them, D and L designation is done in the following way. Starting from the aldehyde or keto group of the monosaccharide, we look at the farthest chiral carbon. For example, in glucose, carbon 1 is the aldehyde. Carbon 2, 3, 4 and 5 are all chiral. The chiral carbon farthest from aldehyde group is carbon 5. So we look the arrange of atoms around carbon 5. If the hydroxyl group of carbon 5 project to the right side, similar to D-glyceraldehyde, this is the D form of glucose. But if this hydroxyl project to the left side of carbon 5, like it does in L-glyceraldehyde, so the given molecule is L-glucose. Now we move to some concepts regarding classification of monosaccharides. Their classification is based on several factors. One important way is to classify them on the basis of number of carbon atoms. For example, a monosaccharide with 3 carbons is called as a triose. One with 4 carbons is called as a tetrose, with 5 carbons as a pentose and with 6 carbons as a hexose. For example, glyceraldehyde has 3 carbons, so it is a triose. Ribose has 5 carbons, so it is a pentose. Glucose has 6 carbons, so it is a hexose. Monosaccharides are also classified based on the carbonyl group they contain. If a monosaccharide has an aldehyde group, it is called as an aldose. If it has a keto group, it is called as a ketose. For example, glucose has an aldehyde group, so it is an aldose. Fructose has a keto group, so it is a ketose. Many a times, both of these parameters are described together. For example, glucose has an aldehyde group and 6 carbons. So it is called as an aldohexose. Fructose has a keto group and 6 carbons, so it is a ketohexose. Glyceraldehyde has 3 carbons and an aldehyde group, so it is also triose. This figure shows the aldose sugars including aldotriose, aldotetroses, aldopentoses and aldohexoses. In this representation only D form of these monosaccharides is shown. You are required to remember the structure of some important of these monosaccharides, which I will notify at the end of this slide. Glyceraldehyde is the simplest aldotriose. It is easy to remember the structure of glyceraldehyde by recalling the structure of glycerol. Glycerol, which is also called as glycerin, has 3 carbon atoms in it. And all of these carbon atoms have 1 hydroxyl group each. If you add an aldehyde group of carbon 1 of glycerol, it will become glyceraldehyde. Phosphorylated forms of glyceraldehyde are important intermediates of glycolysis. Phosphorylated forms of glyceraldehyde are important intermediates of glycolysis. The next are aldotetroses. To the structure of glyceraldehyde, add CHOH, shown in red color, and it will become erythrose, and aldotetrose. Add another CHOH to make it a 5-carbon sugar ribose. Ribose is an important constituent of nucleotides of RNA. Another related aldopentose called deoxyribose is found in DNA. Arabinose is also an aldopentose, but the configuration of atoms on around carbon 2 is reversed as compared to that on ribose. Add another CHOH to aldopentose to make it aldohexose. You can see in this figure that as number of chiral carbons are increasing, the number of isomers is also increasing. You are required to remember the structure of glyceraldehyde, erythrose, ribose, arabinose, glucose, galactose and mannose. On the next slide I will describe how to remember the structure of these aldohexoses. If you remember the glucose, you can use it to produce the structures of galactose and mannose. Glucose is an aldohexose, so first draw 6 carbon atoms. Carbon 1 contain the aldehyde group. Now add hydroxyl group to the right of all remaining carbons except carbon number 3. The hydroxyl of carbon 2, 4 and 5 are on the right side, but the hydroxyl of carbon 3 is on the left side. Now add hydrogen atoms to the carbon atoms. So this produces the structure of glucose. To draw the structure of galactose, I am first going to draw a glucose. Now reverse the position of the hydroxyl group on carbon 4 as well. So the hydroxyl of carbons 2, 5 and 6 are on the right side and those of carbons 3 and 4 on the left side. This is the structure of galactose. For mannose, let us again start with the structure of glucose. This time reverse the orientation of hydroxyl group on carbon 2. So that carbons 2 and 3 have hydroxyl groups on the left side and carbons 4, 5 and 6 have hydroxyl groups on the right side. This is how the structure of mannose looks like. Now let us talk about keto sugars. The simplest keto sugar is called as dihydroxyacetone. You can easily remember the structure of this keto sugar if you know the structure of acetone. Acetone is a 3-carbon compound and contains a keto group of carbon 2. There are 2 methyl group attached to it. If you replace one hydrogen atom of a methyl and add hydroxyl to it, it will become hydroxyacetone group. If you do the same on the other methyl group as well, it will become dihydroxyacetone. Now insert a CHOH group next to the keto group and it will become a erythrulose, a ketotrose. Add one more CHOH next to keto group and it will become a 5-carbon sugar called ribulose, which is a ketopentose. Adding one more CHOH group next to the keto group will produce respective ketohexoses. The structure of fructose is similar to that of glucose except the fact that fructose is a keto sugar. So add a keto group on carbon 2 and replace aldehyde with CH2OH on carbon 1. Rest of the molecule is similar to glucose. Note that the configuration around carbon 3 is similar to glucose. That is, hydroxyl projects to the left side. You are required to remember the structures of dihydroxyacetone, erythrulose, ribulose and fructose. The aldehyde or the keto group present on these monosaccharides can undergo several types of chemical reactions. One such reaction takes place with an alcohol group. The aldehyde group of an aldose sugar can react with an alcohol so that the hydrogen of hydroxyl is transferred to the oxygen of aldehyde group. In this way, the alcohol-containing compound is attached to the carbon of aldehyde. The resulting compound is called as a hemiacetal. Note that this reaction results in producing another hydroxyl group on aldehyde carbon. Now this hydroxyl group can further undergo a condensation reaction with another alcoholic group with the elimination of water. Now there are no free hydroxyl groups on the aldehyde carbon. This is called as an acetal. Similar series of reactions can also take place for keto sugars. The first reaction produces hemiketal. The hemiketal also has a hydroxyl group. This can further undergo condensation to become a ketal. Monosaccharides exist in open chain and cyclic structures. When they form cyclic structures, their hydroxyl reacts with their aldehyde or keto group. Thus aldose sugars upon cyclization turn into hemiacetal. And keto sugars upon cyclization becomes hemiketal. When one cyclic monosaccharide, hemiacetal or hemiketal, undergoes condensation with another monosaccharide to form a glycosidic bond, they are converted into respective acetal or ketal. This figure shows the process of cyclization of glucose and aldohexose. The straight chain form is shown here. But this illustration does not accurately represent the bond angles. We know that when carbon atoms form 4 single covalent bonds, the bond angles are around 109 degrees. Not 90 degrees as shown in the straight chain representation. The next representation shows the bond angles properly. Carbon 1 had aldehyde, so the bond angles are 120 degrees because of a carbon to oxygen double bond. Carbon 1 had aldehyde, so the bond angles are 120 degrees because of a carbon to oxygen double bond. Rest of the carbons are tetrahedral with bond angles of around 109 degrees. So because of these bond angles, the hydroxyl group of carbon 5 is very close in space to the aldehyde carbon 1. So this results in formation of a bond between carbon 1 aldehyde and carbon 5 hydroxyl. This produces the cyclic structure of glucose. Note that the aldehyde carbon of glucose is now a hemiacetal with a hydroxyl group on it. When glucose forms a bond with another monosaccharide through this hydroxyl group, the aldehyde carbon now becomes acetal. This figure shows a condensation reaction between cyclic glucose, hemiacetal, and methanol. After condensation, the bond formed is called as a glycosidic bond. Typically, glycosidic bond is formed between 2 or more monosaccharides. When forming a glycosidic bond, the aldehyde carbon now becomes acetal. The next figure shows cyclization of fructose, which is a ketohexose. Carbon 2 of fructose is a keto group and has bond angles of 120 degrees. All other carbons form single covalent bonds, so bond angles are around 109 degrees. Although we draw the straight chain structure of fructose like this, but the geometry based on actual bond angles is like this. The keto group carbon 2 is in the vicinity of hydroxyl group of carbon 5. This reaction leads to formation of cyclic structure of fructose. The keto carbon is now hemiketal. When fructose forms a glycosidic bond by means of the free hydroxyl, the keto carbon now becomes ketal. Note that although both are hexoses, cyclization of glucose forms a 6-cornered ring and that of fructose produces a 5-cornered ring. You are required to remember the cyclic structures of both glucose and fructose. A chemical compound having a 6-cornered ring with an oxygen atom in the ring is called as pyran. Since the cyclic structure of glucose resembles pyran, so it is called as glucopyranose. Generally, those sugars which form 6-cornered pyran-like ring are called as pyranoses. For example, glucopyranose, mannopyranose, galactopyranose. A 5-corned ring with one oxygen atom is called furan. Those sugars which form 5-cornered rings similar to furan are called as furanoses. Those sugars which form 5-cornered rings similar to furan are called as furanoses. For example, ribose is an aldopentose. It formed 5-cornered ring, so it is called as ribofuranose. Although fructose is a hexose, but it also forms 5-cornered ring, so it is called as fructofuranose. I will talk about the alpha and beta anomers of these sugars next. The carbon atoms of sugars are not planar, which means cyclic structures of sugars are not flat. Because of the bond angles, steric clashes and constraint in the molecule, some atoms of the ring are projected above the ring plane while others project down the plane. This is structure of glucose based on actual bond angles. The carbon atoms are shown in green color. Carbon 1, 2, 3, 4, 5 and 6. Oxygen atoms of the hydroxyls are shown in red color. Hydrogen atoms are not shown here. You can see that when the ring is rotated, all atoms do not lie in the same plane. However, if we look from the side of the molecule, plane of the ring, the position of the hydroxyl group of the hemiacetal or hemiketal may be different. If it project above the plane of the ring, it is designated as the beta form of the sugar. As in this representation, you can see that the hydroxyl on carbon 1 of glucose projects above the plane of the ring, so this is beta glucopyranose. If this hydroxyl projects below the plane of the ring, it is the alpha form. These two alternate forms are called as anomers. Their molecular mass and formula is the same, but their structure is different. The carbon containing this hydroxyl group is called as anomeric carbon. When sugars undergo cyclization, both alpha and beta anomers are possible. As shown in this representation, if the hydroxyl of anomeric carbon projects below the plane of the ring, it is alpha anomer. If it projects above the plane of the ring, it is beta anomer. Both of these anomers are interconvertible as well. Cyclic form of sugar opens up, undergoes rotation around the anomeric carbon and recyclize, thus converting one anomer into the other. In solution, free monosaccharides can exist in all three forms: straight chain, alpha anomer and beta anomer.